Hi and welcome to today's lesson, which is on DNA and the genome. Okay, so for a starter, I'd like you to pause the video and answer the questions that are on the screen, and then we'll go through the answers. Okay, so what is variation? Variation is the differences between living things. We find the DNA in the nucleus of the cell. The structure is a double helix, we'll come on to that later, and you could have said any of these things or other things for things that are inherited, characteristics that are inherited, so eye colour, blood group, attached earlobes, genetic diseases, and so on. Now, DNA itself is a polymer, and what a polymer is a long chain molecule, so it's a very long molecule made up of two strands that form a double helix, and I'll show you a picture of that double helix later on in the lesson. DNA is contained in structures called chromosomes. Interesting word. It's called chromosomes. Chromo means colour, and they were called that because the dyes that we used to stain the cells, when you looked at them under the microscope, made the chromosomes appear as coloured parts of the cell. Okay, moving on. The structure of DNA is made up of long chains of alternating sugar and phosphates. Okay, and attached to each of the sugars is one of four bases. Okay, and that's repeated over and over again. So it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, and each sugar has a base attached to it. Right, so what I'd like you to do is I'd like you to copy and label up the diagram of the structure of the DNA. Okay, so you should have labelled the phosphates as the circles, the sugars are the pentagons, and the bases are the rectangles. And we know that because the sugars and phosphates alternate and the bases are attached to the sugars. Okay, so the structure of DNA, there's that sugar phosphate backbone, and we've got the four bases coming off of that. That makes up the double helix, and this on the left is the diagram showing what a double helix looks like. So it's like a set of stairs going up. Now there are four different bases: adenine, thymine, cytosine, and guanine. And you don't need to remember their names, you just need to know their A, T, C, and G. Now the bases like to hang out in pairs. A and T will hang out together, and C and G will hang out together. Okay, we call that complementary base pairing, and that's really useful for things like DNA replication and making proteins, but you don't need to know any details of that. Okay, so what I'd like you to do is work through this DNA chain and write down what the complementary base pair is for each of the letters. Pause the video for a moment while you have a go at that. Okay, so C goes with G, A, you should have said T, G with C, T with A with T, then we've got a G, an A, two T's, then we've got a C, a T, and another A. If you got that all right, give yourself a big tick. Why is DNA important? Why do we even care about this molecule? Well, the order of the bases in a strand of DNA is what controls our inherited characteristics. The sequence gives a code that produces a protein from little molecules called amino acids. So the order of the amino acids determines what protein your body is going to make. Each set of three bases, we call that a triplet, codes for one particular amino acid. So you need lots of threes to get your full protein. And the bases are held together by bonds between their hydrogen molecules, which we call hydrogen bonds. Okay, so I'm going to show you a little experiment. You can have a go at this at home, maybe if you've...
This experiment, which you can have a go at home, but you will need some help from parents. If you want to have a go at this, you will need some fruit or vegetables. So I'm using frozen peas. You can use dried peas as well. You can also use strawberries, work quite well. I've heard broccoli works and I've heard spinach works. You will need some salt, some washing up liquid, some alcohol. Now, ideally, you want the highest percentage proof alcohol you can get, which would be something like rubbing alcohol. or isopropyl alcohol. I don't have any of that in my house, but what I did find at the back of a cupboard was Benedictine liqueur, which is 40% alcohol, so it should do the trick. Don't drink alcohol. And then I've also got a food processor. If you don't have a food processor, instead of that, you can use a pestle and mortar if you've got one of them, or failing that, a fork and a bowl. And I will explain what you need to do with that later. But the first thing you need to do is you need to put some of your alcohol into the freezer because it needs to be really, really cold for this to work. Okay, so I'm going to do that and I'll be back with you in a sec. Right, when you've got your alcohol in the freezer, the next thing you need to do is put your plant material into your blender or into your bowl, and we're going to add to it a little bit of salt. And we're also going to add to it some water. We want about twice as much water as we have plant material. And what we're going to do is we're going to smush this down or blitz it down until it's a puree. Okay, so when you've made your plant material into a liquid, the next thing you need to do is strain out any solid lumps. So I'm using some fine muslin fabric and a colander to do that, or you can use a sieve, that's absolutely fine. And we just want to sieve that out into a bowl. So what we're after is liquid coming through from the bottom. Sometimes it takes a little while to do that. Right, when you've strained the liquid out of your peas, you can get rid of the solid waste from the peas, and then you need to put your pea juice into a test tube. Now, if you don't have a test tube, like I don't have a test tube, a glass will do the trick. Go for the narrowest glass you can, and you're just pouring the liquid in. Looks delicious. And you really won't want to drink it after this because you're going to add some washing up liquid. You need to add a good squirt of the washing up liquid. Okay, and then you just leave that for about 10 minutes. Okay, so when you've left it for 10 minutes, you then need to get your alcohol out the freezer. And this is the really tricky bit, because what you need to do is you need to very gently pour your alcohol down the edge of the glass. Okay, and what you should get is distinct layers of pea and alcohol. Okay, so if you have a look, you've got the alcohol at the bottom and you've got the peas above it. And if you look in the alcohol layer, there's all these fine wispy bits of white thread. And what those are the DNA from your vegetables or fruit. Okay, so that the alcohol going in has extracted out that DNA. I'm just going to add a little bit more. Okay, and you should see there's a layer forming there where it's got that DNA wispiness in it.
Okay, so looking at genes, gene is one of those sections of DNA on a chromosome, and each gene codes for a particular sequence of amino acids, and the amino acids join together to form proteins. Now, the entire genetic material of one organism is called its genome. We've studied the whole of the human genome now. Have a think about why we might have done that. Well, the reasons are to search for genes linked to different types of disease, to understand and treat inherited disorders, and we use it to trace human migration patterns in the past so we can see where our ancestors came from. Okay, so the key points you need to know. DNA contains bases, there are 3 DNA bases together make a triplet that codes for an amino acid, and the correct order of the amino acids will make one protein. Okay, I want you to have a go at copying and completing this text. Pause the video while you do that, and then we will go through the answers. Okay, so DNA is located in the nucleus of all animal and plant cells. It contains the information needed to manufacture proteins. A long DNA molecule is called a chromosome. Each chromosome is made up of smaller sections of DNA called genes. Each gene codes for a particular type of protein or characteristic. The code within the DNA molecules is made up of 4 bases which pair up and are referred to by the letters A, T, C and G. Okay, have a go at the work I've set you on Fronter, and I'll see you next time.